Hello, everyone. Thanks so much for being here. I know I'm between your lunch, so I just want to share with you that I'm one of the plumbers on this campus, and so we deal with cardiovascular disease. See if I can move my... All right. But before that, I'd like to thank Judy Gasson, as well as Mark, and plus our sponsors for creating these amazing ecosystems in which we can inspire and enrich and empower our next generations. So this project was actually started when I was a student here, looking at this lipids. You can tell the lipids sitting in the middle of the blood vessel. So over the time, we asked the question whether we can identify some specific plaque which is prone to stroke or acute heart attacks. And so the advance of microsensor nanotechnology has enabled our team and to develop smart sensor, otherwise difficult with the conventional precision medicine and precision technology. That technology has been coupled with computation as well as imaging and to improve our clinical acumen. And recently, I think you're probably going to hear some other talk that the presence of microbiota could also be implicated in the modulation of this disease process. So together, this whole collaboration with biomedical scientists, engineers from both UCLA and Caltech, and has resulted in a potential smart sensor to assess unstable plaques. So today, I will probably focus on this microsensor and share with you this possibility of deploying and in the setting of acute angioplasty treatment. So the idea is based on the fundamental electrical impedance spectroscopy, and that fundamental engineering concept has been evolved into a three-dimensional computation, giving rise to better assessment of plaque stability in terms of lipid profiles. And so shown in this slide are the 3D electrodes that can be easily packaged to the inflatable catheters during angioplasty. The idea is to use simple technology but to address complicated clinical scenarios without adding additional fabrication process and manufacturing cost. And I think this process has been continuously garnering interest with the rising use of catheter-based industry. And so our preclinical trial was performed in the pig model, demonstrating the feasibility of this smart sensor to realize plaque otherwise stable or unstable that could be prone to lesions. And shown on the right lower panel is the fractional flow reserve, probably well known to the audience, that has been the cornerstone of our practice as plumbers to detect unstable plaque. But the idea is that if you were a CEO from Kaiser, your job is to cut cost to reduce the number of stents and thereby reducing anticoagulation and the downstream complication. But if you are CEO for UCLA, we're dealing with very prominent people from our neighborhood. You want to be able to identify that plaque, even though these current study indicate a contraindication to deploy the stents. So this is really the marketing undertone for this invention. I think given the time, I just want to share with you that this is well known in terms of the marketing and over time for this type of technology. And this project has been funded through the third cycle by NIH. And also our partnership with Edward Life Science, Boston Scientific, Johnson & Johnson, and we are actually having co-op programs with Edward Life Science to further pursue these directions. Thank you very much for your attention.